So as we discussed that this glycogen primer is formed. Okay, this glycogen primer is formed. We have made. Now then what will happen? Glycogen synthase and an enzyme, it will act on this glycogen primer. This glycogen synthase activity is to use the UDP glucose. UDP, this UDP glucose, it will use a lot of UDP glucose and it will add the glucose in this chain. Like suppose 13 UDP, for example, we have taken, and it will form 13 UDP plus 13 glucose will be attached on this chain. Like this chain will be, this is a glycogen molecule, and this is glucose. A long chain of this will be formed. Okay, this is glycogen. So as we know, between the two glucose molecules there is 1,4 alpha 1,4 bond, glycosidic bond. Okay, we have read in previous classes that it's 1,4 alpha 1,4 glycosidic bond between these glucoses. So as we know that glycogen is a branched molecule. Okay, so now this is a linear, glucose is attaching in the linear form. Now we need someone to make branches. So what will happen? There is an enzyme that is a branching enzyme, that is glucosyl alpha 4,6 transferase. What it will do? This is 1,4 glycosidic bond, but you know in the glycogen molecule there is a branched chain. So when the branching will occur, then here glucose will be formed. Here glucose, here chain of glucose will be attached like this. This every glucose is attached with alpha 1,4 glycosidic bond. Okay, and suppose this is a glycogen molecule. So this branching, this is linear and this is a branching which is occurring. This branching is occurring at alpha 1,6 glycosidic bond. Okay, now how it happened? When there are enough number of glucose, suppose 11 to 12 number of 14 number of glucose are present in this linear chain with the activity of glycogen synthase, this glycogen is there, then what will happen? Then this enzyme which I have discussed, this enzyme glucosyl alpha 4,6 transferase, I'll tell you why this name is there, but this enzyme what will do? It will break the, suppose from here it will break. Okay, it will break it from here and then it will form like this. It will break from here and then it will form here, the branching. So what it is doing, this transferase enzyme, this is breaking 1,4 bond, alpha 1,4 bond, glycosidic bond, and here it is forming alpha 1,6 glycosidic bond. Okay, so it is known as this enzyme glucosyl, because it is transferring the glucose 4 to 6, like from 1 to 1,4 bond, it is transferring to 1,6 bond, alpha 1,4 bond to alpha 1,6 bond, it is transferring. So it is known as glucosyl 4 to 6 transferase. Then after this molecule is formed, then further elongation, here elongation will occur, here more elongation will occur with the glycogen synthase enzyme. When this again, this chain, these chains are again, they are formed above, this glucose chain is above 14, 15 glucose are attached, then this transferase enzyme will again come, it will break and then it will again form branch, and within this enzyme glycogen branched form is formed. So if someone asks you elongation of this glycogen molecule is done by the glycogen, then the answer is glycogen synthase, and if the branching is done by the glucosyl alpha 4,6 transferase. These are the main two enzymes of this glycogen synthesis. Now I'll give you a quick review. Glucose enters the liver through which receptors? GLUT2 receptors. As we know, GLUT2 receptors are the two-way receptors. So, and liver we need glucose to be coming inside and as well as getting outside. So these are the GLUT2 receptors which are taking inside the glucose. Then if there is more glucose, then what will happen? Glucose will be converted to glycogen as and it is stored. Glucose first converts to glucose 6-phosphate through glucokinase, and here the ATP is consumed. It forms ADP. Okay, because we need phosphorus. So ATP to ADP, glucose converts to glucose 6-phosphate through mutase, glucose 1-phosphate, and then this UDP pyrophosphorylase will come. We need UTP over here, and the UDP glucose is formed. as we have discussed all this in previous video. This UDP glucose is formed, and then UDP glucose acts on the glycogenin protein. If the glycogen primer is not present, it acts on the glycogen protein. Here it combines with this glycogenin protein and forms glycogenin with G, okay, and the UDP is lost, and then this glycogen initiator synthase will work on this, and the glycogen primer is formed. Okay, this molecule is formed from here, this molecule from here, and then this enzyme will work and it will form the glycogen primer. Then this glycogen primer is acted upon by glycogen synthase. This was initiator synthase, this is synthase, and then this chain is elongated, so this is a elongation enzyme. It is also known as elongation enzyme. Okay, and then the branching is done through glucosyl 4,6 transferase. As I've discussed, if the chain gets long, then this transferase will break from here and it will add over here. This was alpha 1,4 glycosidic bond, and here the branching, where the branching is done, that is alpha 1,6 glycosidic bond. So like this branching, glycogen molecule is formed.